Dear ladies and gentlemen, my name is Aare JAKSCH and I'm one of the two managing partners at JAKSCH Life Science Consulting. Today we are here to listen to the poster presentation about how to save time and money by smart planning through effective product life cycle management by tailored chemical characterization. The work and the poster will be presented at the Swiss Society of Toxicology at the annual meeting in Basel. Today's speaker is our head of biocompatibility and toxicology. She manages a team of 5 biocompatibility specialists and toxicologists. Charlotte graduated in toxicology and chemistry, and she earned a PhD in organic chemistry and analytical chemistry. She has a distinguished career in medical device biocompatibility evaluations and toxicological assessments, including chemical characterization, spectra interpretation, and study monitoring. Her toxicological evaluations and reviews have saved our clients repeatedly time and money by identifying the most economical but scientifically solid biocompatibility and toxicological strategy. Dr. Charlotte Laupheimer has been the mastermind behind today's presented business case. Ladies and gentlemen, it is a pleasure to welcome my colleague, Dr. Charlotte Laupheimer. Charlotte, please take the stage.
Hi, Aare. Hi, everyone. It's a pleasure to be today here. Actually, I never thought that I got to be as a toxicologist once being Hollywood, but never say never.
Yeah.
All right. Hey, everyone, to you outside there. It's a great pleasure to present today our poster on safe time and money by smart planning and how you can manage your product life cycle with smart chemical characterization. Yeah, and I mean, as you face it also every day in the daily business, this currently ongoing pandemic and its implication has brought the global supply network under pressure. And our clients and manufacturers face challenges due to that because they see regularly at the moment shortage of raw materials, but they face also changes of suppliers, and this brings their product portfolio at risk. Well, you know, we started together that journey by evaluating the existing biocompatibility data set and the evaluations of the established devices. And we had also a look into the physical and chemical data of the new and existing raw materials. And then we drew up a plan, and we wanted to evaluate that material change according to 1093 part 1 and 18. But we decided to evaluate on a raw material level instead of testing, you know, the entire affected medical devices. Then we were allowed to do the study monitoring for our client and had a close collaboration with the testing lab. And then the chemical lab came back to us and they shared with us some preliminary results. And indeed, I mean, it was a beautiful story. Like the FDR spectra showed a high similarity between the existing raw material and the new raw material. So we had like a look into the neat sample comparison, and we also had a look into the non-volatile residues of the extract. And in the end, we had there shown similarity between the two raw materials. Yeah, you know, and we had then also a look into the GCMS data. And as you can see here in the table, we had in the existing raw material actually a higher release in terms of number of chemical compounds, but also in terms of amount. And the new raw material, as you can see in the upper part of the graph, was actually having a lower release of compounds, which is, you know, as a toxicologist, I really love that. However, if you have a closer look, like there were in both materials, there was an unknown compound found. Indeed, they had the same or almost the same retention times, but still, from a toxicology perspective, unknown compounds are a bit challenging. Because in the end, if I have a biocompatibility profile of a existing raw material or of a device, I would like to ensure that this profile is also covering this unknown compound, and I would need to be able to transfer it to the new material. Therefore, we moved on. We asked the lab if it would be possible to do further data analysis and to have a deep dive look into the mass spectra of those two unknown compounds with the very similar retention time. And indeed, they were able to compare the mass spectra, and the mass spectra overlap was very high. We had a very high similarity. And therefore, in the end, we actually came to the conclusion that those two unknown compounds in these two materials are most likely the same unknown compounds. So, and after data collection, of course, we have to analyze them. So we are toxicologists, so we love to do toxicological risk assessment according to 1093-17 and according to ISO TS 21726. And we have seen then a release on a raw material level. And of course, we have to extrapolate it up to the worst case, a medical device article. And then we would compare this release to the TTC according to ISO 21726. And indeed, our release was below that TTC. Therefore, we could conclude that we have not a toxicological concern in this application. Yeah, and after we have completed our toxicological risk assessment, we still moved on with our evaluation of the raw material change. And as you have seen now, we were able to claim chemical equivalence on the testing data. We were also able to show physical equivalence based on literature data. And therefore, we could claim material equivalence between new and existing raw material. And as those two are also used in medical devices with the same intended use, we have also endpoint equivalence. And those two go along, and then you would be able to claim biological equivalence according to ISO 103-18 Annex C. After this, of course, the beautiful story is that then you are able to transfer your biocompatibility profile of your existing clinically established medical devices towards the new proposed devices with the new raw materials. As a chemist by training and also as a toxicologist, I have to tell you, I'm totally in love with chemical characterization. Because in the end, why is it such a beautiful story? We were able to complete ahead of schedule. We were able to save time and money by avoiding further biocompatibility testing, and also, for me, really important, to save animal lives. And therefore, if you have also such challenges, please reach out to us. We are very happy to help you and to discuss with you your situation and to tackle your challenges together.
Charlotte, thank you very much for your insights into this business case on how to save time and money by smart planning and choosing the right toxicological business partner. Thank you to all of you watching us today. We hope you found it interesting and exciting. If you want to learn more about this topic, please download the poster and recording via the provided link. If you want to learn more about the services of the JAKSCH Life Science Consulting, feel free to contact us. Thank you very much and goodbye. Bye-bye.
Ciao.